Hi, welcome to our channel of IGNOU Audiobooks, Indira Gandhi National Open University, School of Sciences, SOS, Master of Science, Zoology, MSCZOO, Semester 2, MZOE002 Animal Behavior and Animal Welfare Ethics. Block 1 Introduction to Animal Behavior, Concept and Evolution. Unit 2 Mechanism of Behavior, 2.1 Introduction. Our present understanding of the mechanism of behavior stems from the pioneering experimental work done by biologists in the 20th century to analyze animal behavior. This approach is known as ethology and was defined by Tinbergen as the biological study of behavior. Tinbergen, 1963. Nico Tinbergen's 4 questions, namely causation, development, function and evolution, inspired by the British biologist Julian Huxley, explain the way in which an animal develops a particular behavior. Of these, the first question explains the mechanism of the functioning of behavior and includes the neural control of behavior through various external and internal stimuli or triggers. It also elucidates the sensory processes and perception through which behavioral output is channeled. The internal triggers that form the source of any behavior are called motivation. Thus, motivation is driven by the external factors together with the physiological requirement of the animal, and environmental stimuli, which is explained by the oversimplistic, psychohydraulic model of motivation, as proposed by Konrad Lorenz. In order to fulfill their needs, animals have evolved complex systems of I. Sensory cells, that perceive changes in the environment, E. A complex nerve system, that transmits and integrates information from sensory receptors, E. Muscle cells, which support the nervous system in translating information into actions, and IV, hormones and chemical messengers, that transmit information for integration and storage. This system interacts to manifest the necessary behavior. Thus, through simple experiments on intact animals or plain observations and combining ethology with neurophysiology, several advances were made in understanding the mechanisms and manifestation of behavior in the last few decades. Objectives. After studying this unit, you should be able to explain the role of nervous system in forming behavior, comprehend the complexity of nervous system results in manifestation of complex behavioral patterns, describe how the characteristics of reflex arc are seen in complex behavioral phenomenon, define sensory ecology, determine the significance of sensory perception, enumerate different types of sensory receptors and their role in animals, comprehend how each sensory receptor is evolved to meet the needs of the animal, describe the concept of primary and secondary orientation, define kinesis and differentiate between its types, define taxes and comprehend its various types, and appreciate the importance of studying mechanism in order to answer the ultimate question in behavior. 2.2 Neural control of behavior. 2.2.1 Relationship between complexity of nervous system and behavior. Primitive animals such as the protists, that possess only rudimentary sensory cells, also show simple orientation behavior pattern. However, behavioral sophistication increases with neural complexity. Let us first recapitulate our knowledge of the neuron structure and its function. The main body of the neuron, called soma, is a broad structure and contains the nucleus. 2 types of cytoplasm-filled extensions called axon and dendrites project out of this soma. Dendrites receive the electrical impulses from the other neurons and pass them on to soma. These impulses are then transmitted from the soma to muscle fibers or other neurons by the axons. Soma, axons and dendrites are all surrounded by glial cells. These cells form a more or less complex sheath around the axon and differ with the complexity of the organism. In invertebrates, the glial cells form a loose, multilayered cytoplasm-filled sheath. Such an axon is called a tunicated axon. Glial cells in vertebrates are known as Schwann cells. They form a tightly bound sheath, where there is no gap left for cytoplasm. Each Schwann cell covers about 2 mm of axon. Between the 2 neighboring Schwann cells of the sheath, small gaps known as the nodes of Renvier are present. At these gaps, the axon membrane is exposed to the extracellular medium. Such a sheath with intermittent Schwann cell arrangement greatly enhances the speed and quality of electrical impulse conduction. These axons are called myelinated or modulated axons. Action potential in a neuron is a all or none phenomenon, where the efficiency of the information is measured by their frequency of generation rather than their graded strength. Except in the sensory receptors and certain other specialized cells, where only a short distance communication takes place. The speed of conduction of action potential across neuron largely depends on the efficiency of axon conductance. Lower animals such as earthworms increase this efficiency by increasing the diameter of axons, called as the giant axons. However, this design may not be cost-effective in terms of materials and space, and hence axons in larger or more complex animals. such as insects and vertebrates, are insulated instead. In effect, the current flows only between the nodes of RNVA, making the conduction extremely rapid. For an impulse to be transmitted, adjacent neurons should be in close juxtaposition, rather than forming a continuous syncytium. Such a region of juxtaposition is known as a synapse, a term which also refers to connections between neurons and sensory receptors as well as neurons and muscle fibers. In invertebrate giant axons, where the juxtaposition is very close, electrical communication takes place for nerve impulse transmission. However, in the more complex animals, transmission across the synapse occurs through various transmitter substances such as acetylcholine, most prominent substance, noradrenaline, dopamine, serotonin, gamma-aminobutyric acid, GABA, and glutamic acid. Such synapses make the nervous system of the animal very efficient and in organizing complex cross connections between neural pathways, making them capable of sophisticated behavior patterns. Advanced nervous system typically consists of 5 components. I. Sensory receptors. E. Afferent or sensory neurons that transmit impulses from sensory cells. E. An efferent or motor neuron that transmit impulses to effector cells. IV. An interneuron linking the afferent and efferent neurons. And V. Effector organ. Such a nervous system is organized most simplistically in cnidarians, such as hydra, jellyfish and sea anemones, and echinoderms, such as sea cucumbers, starfish and sea urchins, in the form of nerve nets, Fig. 2.1. However, as the impulse conducted in such a nervous system is slow, such organisms are capable of simple stereotyped behavior such as fixed action pattern, FAP, and rhythmic actions. Look at the screen. Fig. 2.1. Nerve nets in cnidarians. Centralization of nervous system in its simplest form is seen in flatworms. Central process permits the neural switchboard mechanism, where sensory receptors perceive information and transmit it to afferent nerve fibers which plug into the neural switchboard, connecting them to the relevant effector cells through motor neurons. Central nervous system, CNS, along with the linear organization of ganglia and nerve cord bring about series of behavioral changes in the animal, of which discriminatory abilities and accordingly, specific responses to key stimuli are prominent. Somatic ganglion plays a considerable role in independence of control in higher invertebrates such as feeding, movement, burrowing and copulating behavior in annelids and coordinated movement of legs in cockroaches. However, memory and retention is seen only with enlargement of the brain or cerebral ganglion. Amongst vertebrates, centralization and differentiation of nervous system, as well as concentration of nerves at one end of animal's body, cephalization, is developed in an advanced manner. The brain develops 3 major regions, I, the forebrain or prosencephalon, E, the midbrain or mesencephalon, and E, the hindbrain or rhombencephalon. Each of these are further divided into various regions and control different behavioral characteristics of the animals, ranging from simple behavior such as sensitivity towards environment, heat, pain, light, orientation, etc., to complex aspects such as sleep, aggression, conditioning and decision-making. 2.2.2 Reflex action. Degree of sophistication in structure and organization of brain and nervous system only provides the broad overview of the complexity of behavior an animal is capable of. However, in order to understand the precise neural pathway complexity that controls a certain behavior, let us take an example of the simplest form of behavior, reflex. Reflex may be defined as a rapid, automated response of the whole body or a body part to a simple stimulus. The classic example of a reflex is the knee-jerk response to a stimulation on the tendon of patella. The nerve path traced by the reflex behavior is known as a reflex arc, Fig. 2.2. The simple knee-jerk reflex forms a monosynaptic arc, connecting only a single synapse, where sensory neuron connects directly with the motor neuron. In contrast, a complex reflex response forms a polysynaptic arc, where the transmission of impulses from sensory to motor neurons is passed on through a mass of interneurons or association neurons in the spinal cord. In such a reflex response, the animal contracts the flexor muscles, flexion reflex, and relaxes the antagonistic extensor muscles of the limb to pull it towards the body. Fig. 2.2. A reflex arc showing the path of a spinal reflex. Look at the screen. 2.2.3 Characteristics of reflex arc and their corresponding complex behavior. Reflex arc has certain characteristics, the principles of which are followed even in complex behavior. I. Latency in response. Latency is a certain delay in response to stimulus and is exhibited by both reflex and complex behavior. For example, when a dog encounters a painful stimulus, the latency between the encountering of the stimulus and showing of flexion reflex lies between 60 and 200 milliseconds. A small fraction of time of this delay is taken for nerve impulses to be conducted along axons, while the majority of the delay is due to the synapses between the adjacent neurons. Another example of latency can be cited from the toad's tongue flip to the escape of a cockroach. Just before the toad's tongue flips out of its mouth, tiny gusts of wind produced by the toad's tongue movement, which can be picked up by the cockroach through its numerous tiny wind-sensitive hair on its cerci. However, the critical gust of wind occurs, on an average, 41 milliseconds before the tongue starts emerging from the mouth, while the latency period between the puffs of air and the escape reaction of the cockroach is 44 milliseconds, which is not sufficient for it to escape. It is often difficult to measure latencies for complex behavior. It has been observed that the stronger the stimulus, the shorter the latency of the reflex response. This helps the animal to react swiftly in emergencies. Some evidences suggest that the same is true for certain complex behavior. Hind, 1960, working on chaffinches, a common European finch of which the male has a pinkish-brown breast, was able to measure the latency through the time lag between presenting various frightening stimuli to chaffinches, fringilla coelebs, and their first alarm calls. He observed that the stimulus which was the strongest produced the shortest latency. Summation. It has been observed that sometimes individual neurons respond only after they have received several postsynaptic potentials and, thus, are able to add up or summate excitation coming either at different times, called as temporal summation, or from different places, called as spatial summation. Classic example of this is provided by Sherrington, 1906, through the scratch reflex of the dog, Fig. 2.3. When an irritating stimulus is elicited in the dog's back, the hind leg on the same side is brought forward and is rhythmically scratched at the spot. First, weak stimuli are given singly at 2 points of the skin A and B. They do not evoke the reflex. However, when both the points are stimulated simultaneously, the reflex appears with a latency of about 1 second. Thus, neither is strong enough alone to evoke scratching, but are effective when given together. Summation in complex behavior which occurs between different kinds of stimuli, which may be perceived by different sense organs. For example, A. The sight and smell of food along with the sound of utensils summate when we are hungry. B. Male rats respond sexually to a combination of visual, olfactory and tactile stimuli from a receptive female. Generally, young male rats do not respond unless any two such sources are available. However, mature males, with previous experience, will respond to one type of stimulus alone, which is a case of temporal summation. Warm-up, also called facilitation or motor recruitment. At first some reflexes do not appear at full strength. But, even with no change to the stimulus, their intensity increases over a few seconds. Such types of reflexes are said to be warm-up, where neurons show synaptic facilitation, each successive postsynaptic potential being larger than the previous one. Fig. 2.3, Scratch reflex of dog. Look at the screen. A classical example of this is when chaffinches encounter an owl, they show a type of warm-up effect. The bird gives out a mobbing call in response to the owl. The number of calls given by the chaffinch after the owl is shown is counted in successive 10-second periods. It was observed that the bird begins by calling at a relatively low rate and the maximum calling rate is reached at about 2 to 5 minutes, after which it gradually declines. Similar type of warm-up effect is also exhibited by a pet dog when it encounters an auditory or olfactory stimuli of any unwanted trespasser. Initially the dog responds with slow barking, the intensity increases and then gradually declines. Sherrington showed that such warm-up in some reflexes is due to summation of stimuli which evokes a response from more and more nerve fibers, producing stronger concentration. This phenomenon was named by Sherrington as motor recruitment. Warm-up effect also occurs with complex behavior, not only in the change of intensity but also in the nature of the behavior. This was amply exemplified by Sherrington, 1917, from what he named as the cat's pinner reflex, where at first, repeated tactile stimulation to the cat's ear causes it to be laid back. If the stimulation persists, the ear is fluttered, subsequently the cat shakes its head and, when all the above fails to remove the irritation, it brings its hind leg up and scratches. It is clear that here a more complex behavioral mechanism is involved than the recruitment of a few extra motor neurons. Inhibition. Within the nervous system, nerve cells actively inhibit each other's transmission of information. At the behavioral level, prevention of one's activity occurrence, while another is in progress, constitutes inhibition. Muscles are generally arranged in antagonistic pairs, such that, when one flexes, the other is extended. Sherrington showed that, when in one member of a muscle pair, excitation occurs, it is accompanied by inhibition of its antagonistic pair. However, such inhibition is not absolute. Once a muscle is stretched by its antagonistic pair, then its own stretch reflex will tend to make it contract. Mutual inhibition allows them to take the lead in turn during the limb movements and to alternate flexion and extension of the limbs. In complex behavior, the role of inhibition is less obvious than that of excitation. When an animal is stimulated, the obvious result is that it makes a response. Sherrington showed that the muscles, whose action is common to several different reflexes, compete with each other for the final common pathway. The different controlling patterns of complex behavior such as fighting, feeding and sleeping compete for the control of the animal's musculature. However, only one behavior can occur at a time. The nervous system will allocate priorities since there will often be conflicts as to which stimuli should an animal respond to. Thus, inhibition of action would be as crucial as excitation. This was substantiated by experiments by McFarland. It is pertinent to note that the bird spent only a few seconds on each of these activities. Thus, sustained bouts of each activity in turn inhibited the other, or, in other words, there was alternate inhibition of one activity by the other. Inhibition can be removed through a reflex, which comes back at a higher intensity than it had previously. This is called reflex rebound. For example, when mating activity is not allowed for some time, it has a lowered threshold, and when, at last, it was evoked, it was performed at a much higher intensity. Fatigue. In a reflex arc, the response of a contracted muscle shows a rapid decline despite repeated stimulus. This happens because during repeated stimulation, interneurons of CNS block impulse transmission by increasing the synaptic junction resistance. However, fatigue reflex can be restored by providing a higher or qualitatively different stimulus. A parallel mechanism in complex behavior can be seen as habituation. Habituation is a form of non-associative learning and has been called the simplest form of implicit learning. Habituation refers to a decrease in responsiveness due to the presentation of repeated stimulus. When a snail is presented with the touch stimulus repeatedly, after a few times, it will cease to respond to the stimulus. However, it resumes the response when there is any change in the intensity, spatial or temporal change in the stimulus. 2.3 Sensory processes and ecological perception. Environment has a large amount of information. However, a large part of it is not relevant to the animal. CNS, along with specialized sense organs, work together to not only perceive the necessary information in great detail, but also filter out the rest of it. Sensory ecology is defined as the study of the mechanism with which organisms acquire, process, and respond to information from their environment. For perceiving each kind of relevant environmental cue, there are specialized sensory receptors as follows. 2.3.1 Visual perception. Single-celled organisms and cnidarians perceive only light intensity, which is sufficient for them to thrive in deep water. However, in higher animals, organs for visual perception become more sophisticated. Platyhelminthes and some annelids possess simple forms of eye, placed in a socket, that are capable of large changes in light illumination. The complexity increases by formation of ommatidia, that are grouped together to form a compound eye in polychaetes and arthropods. Although image resolution formed with such compound eyes is poor, they are highly adapted to perceiving even the slightest movement in their surroundings. The latter are well known for their remarkable discriminatory abilities and behavioral superiority, despite a relatively simple nervous system. Thus, compound eyes are clearly beneficial in determining the shape, pattern and movement of things, and in effect distinguishing between food sources, even while flying. The most evolved form of organ for visual perception occurs in vertebrates. Vertebrate eyes are sophisticated organs with multiple cell types and tissue components. The tissues include retina, cornea, lens, iris, ciliary body, Schlemm's canal, circular lymphatic-like vessel in the eye, trabecular network, ocular glands and optic nerve. Of these, the retina forms the most important component, while the other components function on focusing the light on the retina and transmitting information to the brain. The inner layer of the retina consists of two kinds of cells: rods and cones. These two cells are very different in their structure and function. Rods provide poor image resolution and no color discrimination but are highly sensitive to low-intensity illumination. In contrast, cones respond only to high light levels, but provide high-resolution color image. Cones are essentially concentrated in a central region of the retina, the area centralis or macula lutea, where detailed visual perception is most acute. Many animals also possess a depression, which is devoid of blood vessels, in the area centralis of the eye. This depression, called fovea, enhances the focus of vision. Birds possess two foveae, the second one situated at the back, which helps them to discriminate objects during their flight. Ganglion cells in the retina show a remarkable diversity in function, which aids the brain in excluding unnecessary information from the surrounding and perceive only the biologically relevant ones. The functional discrimination of these cells varies with species. For example, retina of frog has 5 types of cells that can perceive information related to food, predators or mates. In contrast, rabbit possesses at least 8 types of retinal cells that respond to varied stimuli such as speed and direction of movement, stimulus orientation and stimulus absence, as well as contrasts in object borders. Retinal cells of sophisticated insects such as honeybees possess a trichromatic system of retinal receptors which respond to ultraviolet, blue-violet and green-yellow spectrum. In contrast, humans can perceive color spectrum between 400 to 700 nanometers. Thus, each animal's vision is adapted to its specific needs. 2.3.2 Auditory perception. Sounds are produced through vibrations emitted from a variety of sources in the environment. These vibrations are in wave-like motion and cause pressure changes. Organs for sound perception have evolved to capture these vibrations and pressure changes. Although many lower animals perceive pressure changes or vibrations, insects are the lowest forms with a defined organ for auditory perception. Many insects possess tympanic organs, located on the legs, thorax or abdomen, which contain a tympanum connecting to a series of sensory neurons. In insects such as crickets and grasshoppers, tympanum vibrates due to the vibrations from sound waves, stimulating the auditory nerve to send impulses to the CNS. Similar to retinal cells, the sensory neurons of the tympanum are grouped based on their response to certain sound wave frequencies. This permits the insect to discriminate information from the sound reaching its ears. During the course of evolution, the complexity of ear structure and the range of information it can convey has become increasingly complex and sophisticated. In mammals, the ear is divided into I, pinna or the external ear, pinna, E, middle ear, and E, inner ear. The cartilaginous pinnae receive and conduct the sound waves into the auditory meatus or the ear aperture. The external and middle ears inside the ear aperture are separated by the tympanic membrane or eardrum. Vibration of the tympanic membrane causes the first, malleus, of 3 bony ossicles to vibrate, which, in turn causes vibrations in the other 2 ossicles, i.e. incus and stapes. These vibrations cause an inward bulging of fenestra ovals of the cochlea in the inner ear. This pushes the cochlear fluid, endolymph, causing an outward bulging of fenestra rotunda in the cochlea, thus moving cochlea back and forth. Special hair cells in the inner ear transmit the impulses caused by these movements to the temporal lobes of the brain where they are translated into sensible information. Similar to retinal cells, auditory cells are also specialized in the type of auditory signals they respond to. For example, nocturnal animals that depend heavily on the auditory stimuli for locating prey have evolved specialized mechanisms to locate a source of sound. The sound that they perceive is highly accurate and serves their specific purpose. For example, in owls, orientation of the auditory meatus and the manner in which their facial feathers are arranged only permit sounds along their line of vision. Moreover, the position of their ears on head is not uniform. This helps in assessing the location of the sound more efficiently. Thus, they simply orient their head in the direction of sound to determine its source. Fig. 2.4. In contrast, although insects respond to a highly limited range of sound frequencies, it serves their purpose of finding a mate or escaping enemies. For instance, male mosquitoes perceive the low-frequency vibrations that the wings of female mosquitoes produce. Similarly, the ears of moths of family Noctuidae have evolved to entirely focus on the intensity and pattern of emission of vibrations produced by bats. These moths possess 2 kinds of neurons in their ears, those which respond to low-intensity intermittent sounds and those which respond to high-intensity sounds. Low-intensity neurons are capable of capturing the faint ultrasonic emissions of bats from up to 10 meters and increase their signaling strength as the sound vibrations from the bat gets closer. The adjustment of sound perceived by the 2 ears helps the moth to determine the exact position of the bat, which helps them to escape it. In contrast, neurons perceiving high-intensity sounds are fired up only when the bat is flying very close to the moth. When the moth receives signals from these neurons, they start flying erratically, thus immediately removing themselves from the direct flying path of the bat. Fig. 2.4. Asymmetrical location of ears aid the bat in detecting the precise location of its prey. Look at the screen. 2.3.3 Tactile perception. A wide variety of animals rely on low-frequency stimuli of touch or pressure for performing various behavioral activities such as mating rituals or parental care. Many species, such as those from class Insecta and Mammalia, possess hair on their body or part of their body that have touch-sensitive nerve. Endings. For example, honeybees orient themselves on the hive using hair present between their head and thorax. Moreover, their communicatory dances also depend on tactile stimuli. Most mammals, including primates, except human, possess special facial hair, called vibrissae, commonly called whiskers, for sensing their environment through tactile signaling. 2.3.4 Other mechanoreceptors. Besides the mechanoreceptors used for auditory and tactile perception, many animals also possess other forms of mechanoreceptors to sense pressure, motion and stretching. Stretch receptors affect behavior of the animals by regulating their posture and locomotion. Similarly, structures such as statocysts in crustaceans and semicircular canals filled with endolymph inside vertebrate ears aid in the positional correction of the animal. Fig. 2.5 and Fig. 2.6. Fig. 2.6. Vertebrate semicircular canal. Look at the screen. 2.3.5 Olfactory perception. Many animals utilize chemicals as their primary means of communication between conspecifics and with other species. In order to convey information within their own species, animals use pheromones, which are different from allomones, used for interspecific communication, and hormones, chemicals functional within the animal's own body. Pheromones are used by many mammals, such as members of canine and feline families, to mark their territory. A classic example of mechanism of functioning of pheromones is perception of bombicle, the sex attractant pheromone of silk moth Bombyx mori. The receptive female moth secretes bombicle in small amount in the air, which travels downwind. Even as little as 200 molecules of these pheromones are sufficient to be detected by the long feathery antennae of the male moth and help them to orient towards the female. Other females cannot perceive these pheromones. However, they can detect the olfactory signals from surfaces that could be potential oviposition sites. Thus, olfactory receptors of various animals perceive different kinds of information such as sex, age, and motivational state of the emitter, which aids them to modify their behavior. Accordingly, human olfactory system is depicted in Figure 2.7. Look at the screen. Fig. 2.7. In the human olfactory system, bipolar olfactory neurons extend from B, the olfactory epithelium, where olfactory receptors are located, to the olfactory bulb. 2.3.6 Gustatory perception. Olfactory receptors are often interconnected with gustatory or taste receptors. However, their functional mechanism is quite different. While olfactory receptors are mainly restricted to antennae, in invertebrates, or nasal cavity. In vertebrates, gustatory receptors are found at various locations in the body of different animals. In flies, these receptors are found in mouth parts and legs, and fire in response to water, sugar and salt. Fig. 2.8. Vertebrates have these receptors within the mouth. In human, in particular, gustatory receptors, or taste buds, are found all over the surface of tongue. A popular myth is that human tongue has well-defined gustatory receptors for different tastes. Fig. 2.9a. However, recent studies have shown that all regions of the tongue can perceive all tastes, though some regions are more sensitive to a particular taste. Fig. 2.9b. The chemical nature of the substance being tasted provides the animal information about its nutrition value, as well as a possible harmful effect on its survival. Thus, many behavioral decisions such as avoidance or preference, and the possible ensuing conditioning behavior, are made by the animal based on this perception. Look at the screen. Fig. 2.8a. Location of gustatory receptors on A. Insect body parts and B. Head region are shown with yellow highlighting. C. Diagrammatic representation of Lepidopteran caterpillar head. Fig. 2.8b. The position of the gustatory organs, wings, tarsi, labella, and pharyngeal organs, in Drosophila. Fig. 2.9a. Tongue map according to popular belief. B. Taste receptor cells. Often, mechanoreceptors and chemoreceptors are found to be associated with each other in order to aid the animal in not only perceiving the chemical cue, but also in detecting the location and direction of its source, and orienting itself accordingly. For example, the hair on the mouth parts and legs of the blowflies, Phormia spp, have 5 afferent sensory neurons, of which one detects mechanical stimuli, while the other 4 detect gustatory stimuli responding to water, sugars and salt. Other than the mechanisms discussed above, animals are known to perceive other cues such as temperature, electric current and magnetic forces. All these mechanisms play a direct role in sensing the environment surrounding them and form the most important step in the process of filtering out meaningless information. 2.4 Mechanism of orientation. 2.4.1 Primary vs secondary orientation. Orientation is the alignment of the animal with reference to a particular stimulus. This is the position that the animal needs to maintain, achieve in order to reach the source of the stimulus or in order to reach satiation. Orientation is defined as the capacity and activity of controlling location and attitude in space and time with the help of exogenous, external, and endogenous, internal, stimuli. Gender, 1963. This behavioral aspect is particularly significant for any organism in order to transfer from inhospitable to hospitable environment. Organisms are constantly under one or both of the following forms of orientation. A. Primary orientation, the default body position in space either in a stationary or in a moving position, i.e. the normal posture either in a stationary or in a moving organism. B. Secondary orientation, the directional alignment of the organism in response, towards, away or at an angle, to a particular stimulus. Secondary orientation is superimposed on the primary orientation. Secondary orientation relies on two distinct patterns of movement, I. Kinesis. E. Taxis. 2.4.2 Kinesis. Kinesis is the non-directional and random movement of an animal in response to stimuli. This response is relational to the intensity of stimulus. In many animals, the speed of locomotion changes with the change in the intensity of stimulus and the accumulation of kinetic energy in the animal. This form of kinesis is called orthokinesis. For example, with the decrease in humidity in the environment, the speed of movement of wood lice increases in an attempt to escape from the low humidity area. Similarly, cockroaches move away from the source of light in search of darker areas. Some animals change the direction of their movement based on the intensity of a stimulus. This form of kinesis is termed as clinokinesis. Generally, it is seen in animals with a single receptor, because of which they show an alternate left-right-left movement to compare the direction and intensity of the stimulus for obtaining accurate orientation. Insect caterpillars and maggots show such a movement while searching for a place to undergo pupation. Moreover, different types of kinesis are termed based on the type of stimulus, e.g., chemokinesis is kinetic movement with respect to chemical stimuli. Photokinesis is based on the gradient of light, etc. 2.4.3 Taxis. Taxis is the animal orientation, movement with respect to direction of the source of the stimulus in space. This orientation, movement may be towards, away or at an angle from the stimulus, according to the perceived stimulus. It can be divided into various types such as phototaxis, light, thermotaxis, temperature, hygrotaxis, humidity, chemotaxis, taste, odor, geotaxis, gravity, anemotaxis, air current, rheotaxis, water current, menotaxis, angle to the stimulus, etc. Some of the more complicated types of taxis are described below. A. In clinotaxis, the animal compares the intensity of stimulus by alternate lateral movements of part or all of the body. Such an orientation occurs in organisms such as euglena, which have a single receptor. B. In contrast, tropotaxis is found to occur in organisms which have paired receptors and hence get equal inputs, enabling them to move in straight line, such as eyes of planaria. C. Teletaxis occurs in animals when they are given a choice between the two stimuli. This type of orientation is seen in honeybees and is achieved by making a choice and moving towards any one stimulus. D. Menotaxis involves orienting at an angle to the source of the stimulus. For example, during waggle dance, honeybee scouts keep changing their angle of straight run in accordance to the movement of sun. This is memorized by foraging bees while watching the dance on the vertical surface of the comb. This helps the foragers to fly from their hives to the flowers by maintaining a constant angle to the sun. Thus, this kind of orientation is also commonly called as compass orientation. A. Nematoxis, memory movement, was studied extensively by Nico Tinbergen, 1951, through his work on digger wasps. Before leaving for foraging, wasps circle around the nest and memorize the map and landmarks surrounding the nest. This aids in accurately orienting themselves when they return to the nest. Many other insects, such as ants, use memory to follow trails that their nest mates have left when traveling. 2.5 Summary. Let us summarize whatever you have learned so far. All behavioral patterns in animals, whether innate or learned, are developed through a complex mechanism that includes receiving information about its surrounding environment through various methods of communication, processing and associating it in the central nervous system, and forming a response mechanism is one of the proximate question asking how a certain behavior is performed and what intricate components of the body coordinate with the surrounding environment in order to develop an appropriate response. Understanding the mechanism of behavior thus provides a better insight into the ultimate question of why such a coordination was evolved in order for the behavior to take place. Reflex may be defined as a rapid, automated response of the whole body or a body part to a simple stimulus. Latency in response, summation, warm-up, inhibition and fatigue are characteristics of reflex arc. There are specialized sensory receptors for visual perception, auditory perception, tactile perceptions, olfactory and gustatory perception. Orientation is defined as capacity and activity of controlling location and attitude in space and time with the help of exogenous, external, and endogenous, internal, stimuli. Orientation is of two types: primary and secondary. Secondary orientation relies on two distinct patterns of movement, i.e. kinesis and taxis. Kinesis is non-directional and random movement, whereas taxis is movement with respect to direction of the source of the stimulus in space. Thank you.